So now, first of all, that is 5' cap formation. So if you see that this one, that these are the various stages or the steps which occurs during the co and post transcriptional modifications in mRNA. So first of all, that is 5' cap formation. After that, transcription termination. Then 3' end processing and polyadenylation, splicing, editing, nuclear export of mRNA, and as well as the mRNA stability. So all these, nothing but all these are the stages which are occurring during the co and post transcriptional modifications in mRNA. Okay, now we will move with this slide. Just take the screenshot of this slide so that after this lecture you may read at your home. There are some elongation factors are there, that is HS55 and the P80SF1. These factors, these elongation factors will help during the elongation, and when the elongation start means in last lecture in our campus when I conducted that lecture, at that time we learned that there was an initiation. Initiation is what? That the RNA polymerase will bind over the promoter region, right, or over the DNA, then it will start the transcription. Now the actually addition of the ribose sugars in a particular strand, it is called as the elongation. So for that elongation, for the initiation, there are some certain factors are required, that is general transcription factors, right? Transcription factor 2B, A, B, F, E, H, right? Likewise, here also there are certain elongation factors are there which are called as the HS55 and P80SF1. One by one, we will learn the role of these elongation factors. But here, according to our third point, it is said that it is given in text that particularly these elongation factors will help during the elongation, means for the elongation, termination, as well as for the processing of RNA. Now, the processing of the RNA, it means that splicing mechanism is going on. Okay. Now, over the 5' end of the RNA, what I said that there is a methylated guanine will be added over the 5' end. And this methylated guanine, particularly that 7th position of the guanine, we will learn here after this slide, that 7th position of the guanine will be methylated, methyl group will be added over that along with one enzyme, with the help of an enzyme, and this methylated guanine will be attached over the 5' end of the RNA. At that time, see the last point, that there will be the unusual, means usually this does not occur this condition, but unusual 5' linkage in between the 3 phosphate groups. So first of all, it is, let's see that this one, this is the RNA or mature RNA. This is the 5' UTR, means 5' untranslated region. And this one, particularly this one, this is the 7-methyl guanosine. And this 7-methyl guanosine will added over the 5' base or 5' region of this mRNA. Now, how this capping formation, or how this capping process occurs, we will hear one by one. First step, that this phosphate group will be removed. You see here, particularly this one is the first phase of the RNA. These are the 3 phosphate groups. Last time I have explained that we will designate that alpha, beta, and the gamma phosphate. So out of this, the phosphate group will be removed with the help of this enzyme, that is RNA triphosphatase. We know that the phosphatase always remove the phosphate group. So in this case also, that one phosphate group will be removed. You see that this is the RNA and this is the RNA triphosphatase. So this enzyme will remove the one phosphate from this RNA. So here only 2 phosphate groups are there, alpha and beta. So this is the first step during the 5' cap formation, in which that one phosphate group is removed from the 5' end of the mRNA. In second step, that GTP is added with the help of guanylyl transferase. So overall, means obviously one slide is remaining there. So in this step, you have to just remember that there are 3 enzymes are there which are required particularly during the 5' cap formation, and see their roles. According to their role, the question may be asked in your examination. So in first process, in first step, what we learn during the cap formation, that the RNA triphosphatase enzyme is there, which will remove the one phosphate group from the RNA molecule. After that, the another enzyme, that is guanylyl transferase. This guanylyl transferase itself, second enzyme, that is guanylyl transferase, this means what? It will transfer the guanylyl group. So this is the free guanosine. This is the ribose sugar. guanine and 3-phosphate. So the same one, alpha, beta, and gamma. These are the 3-phosphate. So see here that though it is not shown here, this oxygen having the lone pair, the oxygen very soon it will because it having the ability to deprotonate or to undergo the deprotonate. So this oxygen will attack over this alpha phosphate. So see carefully, particularly in this step, that GTP, as it is said that GTP is added with the help of the guanylyl transferase. So this GTP, free GTP will be added over this RNA molecule. Okay. Now, this is the 5' unusual linkage. Now see, if you continuously see here that over this particular end, what you will observe, there is a 3' end of that RNA, right? Here, there will be the 3' end of the RNA. But if you see here on your left side, here again the 3' end is there. There is no any further 5' end is present. There will be only the 3' end. It means that now on this mRNA, there is 3' end here, and again there is a 3' end. Means both end having the 3' end, right? Now, in next step, in third step, the nucleotide means whatever the nucleotide that is GTP which was added over this mRNA, it is modified by the methyl group because the nuclease may attack over this nucleotide to show this unusual base that the next enzyme that is methyl transferase, this methyl transferase will transfer the methyl group over the 7th position of this guanine. Now it becomes the 7-methyl guanosine. Now it acts as a cap for this mRNA. It means that further any, because already this nucleus, this mRNA is present, it is located, still it is located within the nucleus, and within the nucleus there are number of nuclear nucleases are present, or nuclear enzymes are present, and anytime they may attack over this RNA and they may destroy this RNA. That's why to protect this RNA from the enzymes, this is nothing but this unusual base that is 7-methyl guanosine is added over the 5' end of this RNA. Now this unusual base act as a cap. That's why the full process is known as or called as the capping process. Now this RNA, when the RNA, last time we have learned the structure, particularly this is the RNA polymerase and this is the DNA. This is the C-terminal domain or CTD. RNA is capped when it is only 20 to 40 nucleotide long. This RNA chain is very short, and when it is 20 to 40 nucleotide long, at that time the capping process will occur. Now, before going to start the splicing mechanism, first of all, capping enzyme is responsible. Now see carefully that this RNA polymerase having C-terminal domain or the tail-like structure or CTD tail. It is also called as the C-terminal domain or CTD tail. Now this CTD tail having some heptapeptide amino acid sequences over there. Okay, last time I explained that. We'll go particularly here. Now see that particularly YSP, PS, PS. In that particularly 7th position of this serine and 5th position of this serine. So if there is a phosphorylation occurs on the 2nd position, what will occur? It is given here, and again given in a previous slide also. And if the phosphorylation occurs on the 5th serine, then which changes will occur and what will occur here? It is given here. 7-heptapeptide. It repeats near about 52 times in mammals and near about 26 times in yeast. I already explained that the RNA is capped when it is only 20 to 40 nucleotide long. Then phosphorylation of the serine, which is present at the 5th position. So if there is a phosphorylation, means if you see that particularly the capping enzyme is attached over here, means capping enzyme is recruited at this particular location. Now this RNA polymerase will move here and it will start the elongation process, right? Now during the elongation, before the elongation, this capping enzyme will recruit the other enzymes which are responsible for the capping process. So one by one we have learned about the 3 enzymes, but 3 enzymes will be recruited by this enzyme, that is capping enzyme. So there is a phosphorylation. So when that capping enzyme will reach here at this particular point or the area, when there is a phosphorylation in that tail region of that RNA polymerase, and which position, either 2nd or the 5th position. So your answer will be the 5th position, because phosphorylation of the serine at the 5th position is responsible for the recruitment of capping factor. So after capping enzymes. So after capping, dephosphorylation serine 5 within that particular 7 heptapeptide repeats. And once the capping is over, it means that this enzyme should be removed from this area so that there should be the dephosphorylation. So after capping, dephosphorylation of serine fifth position serine within the repeats leads to the dissociation of capping machine. So this capping machine or the enzyme will be dissociated from this area when there will be the dephosphorylation. Then the next step, that will be the phosphorylation of the serine, which is present at second position. So this phosphorylation of second serine present at second position, it will recruit the splicing factor. So see here that it was the phosphorylation of fifth position. Now it will again undergo the dephosphorylation, and after that the phosphorylation at the second position of that serine. So if there is a phosphorylation of the second serine, it means that it is responsible for the recruitment for the splicing machinery or the splicing enzymes which are required for that particular step. Is given here. First of all, phosphorylation leads to the recruitment of fifth prime splicing machinery, and the next one is that after capping there is a dephosphorylation. Then the second serine undergoes the phosphorylation. So phosphorylation leads to the recruitment of TAG splicing factor. It is also called as the TAG-SF, means splicing factor. So during this process or inside, particularly in this process, you have to just keep it in mind the names of the factors and their roles. Particularly, this TAG splicing factor will recruit the spliceosome machinery on C-terminal domain or CTD. One by one, we will learn this particular mechanism. Before that, the capping functions. So, cap, it will prevent the 5' end of the mRNA from being digested from the exonuclease. Then, this 5' cap not only protect the RNA from the exonuclease, but it will help the mRNA to transport out of the nucleus also. During the transportation, this cap will also help. Next one is that the cap plays very important role in initiation of mRNA translation. Okay, so this 5' cap, it is very important or useful during the mRNA translation, that is protein formation. Then we will learn about the splicing, that how splicing occurs. Two protein complexes, that is CPSF and CSTF, these are carried by the C-terminal domain of the polymerase at its approaches at the end of the gene. One by one, we will learn about these two factors. First one, that is CPSF. CPSF means what? Cleavage and polyadenylation splicing factor. They are responsible for the cleavage and polyadenylation also. So, cleavage and polyadenylation specific factor and cleavage stimulating factor. So, these two protein complexes, they are carried by the C-terminal domain of a polymerase, that is RNA polymerase, at that RNA polymerase approaches or reaches up to the end part of that gene. It means that where there should be the both complexes, that is CPSF and CSTF, they will be recruited or carried along with the RNA polymerase, and they will play their role. How they will play their role, we will learn here one by one. Now, within the DNA, if you see particularly here, that there are certain sequences, they're called the poly-A signal sequences in DNA, right? Because that mRNA is taking all the sequences, complementary sequences from the DNA. It means that these sequences are present within the DNA itself, and these sequences are called as the poly-A signal sequencing DNA. Particularly, these sequences are located in the terminator sequences or terminator region of that particular gene. Now, the sequences which are transcribed into the RNA trigger transfer these factors to the RNA are called poly-A signals. Now see here that the C-terminal domain, it is already phosphorylated. This will be phosphorylation of the second serine or second number of that particular serine amino acid. And here the two factors, that is CSTF and CPSF, these two factors, they are joined with this mRNA. Now, once CPSF and CSTF are bound to RNA, other proteins are recruited and leading initially to RNA cleavage and then leads to the polyadenylation. Now, these two factors, these are responsible for what? That is for RNA cleavage, means RNA should be cleaved or should be removed or should be separate from this RNA polymerase from this side. And again, the free 3' end of that RNA. Over that, the number of adenine residue should be added. We know that 5' end actually there is no any actual such 5' end is existing over this mRNA. But still, for our convenience, we will say that this is the 5' end of that mRNA, and that mRNA particularly over which that capping is what done. Now the 3' end, which is cleaved from this RNA polymerase, over which that nucleases or nuclear enzymes may attack over this 3' end, and they may cleave this RNA molecule, they may digest this RNA molecule. That's why the other enzyme is there, that is poly A polymerase, which will recruit over this particular area, and it will add the adenine residue. How it will add the adenine residue, we will see one by one. But here, first of all, that once CPSF and CSTF bound to the RNA, other proteins recruited leading initially to RNA cleavage and then leads to the polyadenylation. Polyadenylation is mediated or done by the enzyme that is PAP, poly A polymerase, which adds approximately or near about 200 adenine residue over the 3' end of a particular mRNA, which is produced by the cleavage. Now, this 3' over this 3' end, this poly A polymerase will add the adenine residue. Now, this enzyme, particularly which one, that is PAP or poly A polymerase, this enzyme uses the ATP as precursor and add nucleotides. It is not clear what determines the length of the poly A tail. Still, it is not known. But over this RNA, this CSTF and CPSF, they will bound with this particular poly A signal sequences in the RNA. Then this CSTF, cleavage stimulating factor, this cleavage stimulating factor will cleave this RNA from the RNA polymerase, and then we will get the free mRNA. Now, but still there is a cleavage and polyadenylation specificity factor is still present or attached with these poly A signal sequences of the mRNA. Being that CPSF is present over these sequences over the mRNA, it will recruit what? That is CPSF will recruit the PAP, that is poly A polymerase, and poly A polymerase will add the adenine residue. There is a poly A binding protein. This poly A binding protein will help this PAP to add the adenine residue. So this enzyme, that's why it is given here that this enzyme, that is PAP enzyme, uses the ATP as a precursor and adds the nucleotides. It is not understood what links polyadenylation to term, but it is clear that polyadenylation signal is required for the termination. Basically, the two models have been proposed to explain the linkage between that polyadenylation and the termination. So, transfer of 3' processing enzymes from the polymerase, that it will trigger the conformational change. It is not known that which will determine the length of the adenine residue and how this particular ending process, that cleavage from that RNA polymerase, that RNA will be removed from the RNA polymerase, RNA polymerase will remove from that DNA. Still, this process is not fully known, but there are certain hypotheses. So, first one is that transfer of 3' processing enzymes from the polymerase, it will trigger the RNA into the conformational change in RNA polymerase, and which will reduce the processability of the enzyme, leading to a spontaneous termination. Then the next one is that the second model proposes that the 5' cap on the second RNA molecule is sensed by the polymerase, and which recognizes the transcript as the improper and terminates. See that once the RNA polymerase reaches up to the end region, of the particular gene, it will encounter certain specific sequences that after being transcribed into the RNA, and that these sequences will trigger the transfer of the polyadenylation factors or that particular enzymes to the RNA. As already I told you that transcription termination is two factors, that was CPSF and CSTF. CPSF and CSTF, generally these two factors, they are responsible for the termination of that particular what was the transcription was going on there, because these factors are responsible for the cleavage of that RNA from the RNA polymerase, and without the RNA, that RNA polymerase will not work over that gene. It is said according to that particular model. That's why the RNA polymerase very soon will release that DNA, and obviously indirectly this process is called as the termination. So once the RNA polymerase reaches up to the end part of the gene, it will encounter specific sequences. These are called the poly A sequences. If you remember that, particularly here at this region. So once the RNA polymerase will reach up to these sequences, after that these sequences will be added into that RNA. And once these sequences added into the RNA, this factor, that is CSTF and CPSF, this factor will be recruited in that particular area. And it means that indirectly it is the termination of that transcription process. Then leading to the three events: cleavage of the message, that is RNA. So RNA will cleave from that RNA polymerase. Then second thing is that polyadenylation. So many of the adenine residues will be added, near about 200 adenine residues will be added by the poly A polymerase, this enzyme. And the third one is that is important. The third one is that the transcription termination, which was going on by the RNA polymerase. So RNA polymerase will also leave from that site, from the terminator sequences. So in this way, this was the termination of that transcription.